Okay, so welcome to this next video in which we are discussing the adrenoreceptors. Okay, right. So we've done alpha 1, alpha 2 and beta 1 now. What we now want to turn our attention to is the beta 2 adrenoreceptors. Okay, right. So we'll start off with where is the localization of the beta 2 adrenoreceptors. Well, basically they are also found on smooth muscle cells, just like alpha 1 adrenoreceptors were found on smooth muscle cells. Okay, so I'll draw my beta 2 adrenoreceptor here on our smooth muscle cell. Okay, so here's the amino terminus, here's the carboxylic acid terminus. And basically they trigger the exact opposite thing to the alpha 1 adrenoreceptors. Okay, so this is a beta 2 adrenoreceptor. And basically it, when activated, is going to trigger relaxation of the smooth muscle cell. And you might think, well, this is quite odd, because smooth muscle cells will also have alpha 1 adrenoreceptors on their membranes, which are telling the smooth muscle cells to contract. And both alpha 1 and beta 2 adrenoreceptors have the same ligand. Well, basically, you might be sitting there wondering, well, what's going to happen? Which one is going to win? Well, basically, what determines which one is going to win, i.e. whether your muscle is going to contract or relax upon stimulation by adrenaline or noradrenaline, is the number of alpha 1 adrenoreceptors that you have compared to the beta 2. Okay, so basically there are many different sites where you have smooth muscle cells around the body. Okay, and some of those smooth muscle cells you will want to contract in response to adrenaline or noradrenaline. Okay, so for instance, the smooth muscle cells which surround blood vessels, vascular smooth muscle cells, you generally want those to contract in response to noradrenaline or adrenaline, because generally noradrenaline and adrenaline will be released as part of the sympathetic nervous system onto smooth muscle cells. And the idea then is that in the flight or fight response, you want basically to raise your blood pressure so that the delivery of blood to the tissues is going to be maximal, basically. Okay, and specifically you want to get it to the skeletal muscle. Okay, so you'll want to contract the smooth muscle cells that surround blood vessels to raise blood pressure. Okay, right. There are other examples then, such as the airway smooth muscle, where you would want that to dilate in response to the sympathetic nervous system. Okay, so when adrenaline and noradrenaline are put on bronchial smooth muscle, they cause dilatation of that muscle, and that's so that you open the airway so that the amount of air that you can get into the lungs is going to be increased. Okay, and that's again going to help you in a fight or flight situation. Okay, right. So different smooth muscle cells will have different fractions of alpha 1 and beta 2 adrenoreceptors. Okay, and if you want your smooth muscle cell to contract in response to adrenaline or noradrenaline, you'll have a lot of alpha 1 adrenoreceptors on the surface of it. And if you want it to relax in response to adrenaline or noradrenaline, you'll have a lot of beta 2 adrenoreceptors on their surface. Okay, so we now want to see how beta 2 adrenoreceptors actually trigger relaxation of the smooth muscle cell. Okay, so let's just remind ourselves of how smooth muscle cells contract. Okay, so you have this meshwork within the smooth muscle cell, remember, where we have many of these dense bodies like so. Okay, and these dense bodies will have contractile units between one another. Okay, so for instance you could have contractile units like so. Okay, and basically to activate these contractile units to contract, remember what we need to do is phosphorylate the regulatory myosin light chain on the myosin cross bridge, and then what will happen is these contractile units will contract and the whole smooth muscle cell will contract. Now, to phosphorylate those regulatory myosin light chains, we need the enzyme myosin light chain kinase to become activated. Okay, and remember how you get myosin light chain kinase to become activated is that you increase the level of calcium in the cytoplasm, and then what happens is you get calcium calmodulin complexes forming, okay, and these calcium calmodulin complexes bind to the myosin light chain kinase enzyme, and they will activate the myosin light chain kinase. So here is the calcium calmodulin complex bound onto our myosin light chain kinase. Okay, so basically, beta-2 adrenoreceptors are going to target the contraction of smooth muscle cells in multiple different ways. And I want to stress that this pathway is still contentious. It's in one of those areas that it's still quite foggy. We're not absolutely sure we've got it. We've got a good idea, but it's still a little bit hazy and not very well understood, okay? So I'm going to give you some answers here, but be aware that they may well develop in future years. Okay, so I'm going to give you what the best answer is at the moment. Right, so basically a simple part of the pathway by which you can stop the contraction of the smooth muscle cell by activating the beta-2 adrenoreceptor is basically, we know that the beta-2 adrenoreceptor is going to interact with the GS heterotrimeric G protein, producing the alpha-S GTP subunit, which then goes off to activate adenylyl cyclase enzymes, producing cyclic AMP, which activates protein kinase A. Basically, protein kinase A catalytic subunits can phosphorylate myosin light chain kinase. Okay, and basically when you phosphorylate myosin light chain kinase by protein kinase A, so it's protein kinase A that does this, okay, specifically the catalytic subunit of protein kinase A, I should probably put that, catalytic subunit of protein kinase A. Basically, myosin light chain kinase now becomes insensitive to the calcium calmodulin complex, so even though the calcium calmodulin complexes come and bind to the myosin light chain kinase, it doesn't become activated by the calcium calmodulin complex, so it then doesn't phosphorylate the myosin light chain, specifically the regulatory myosin light chain, and you don't get activation of the myosin cross bridges, which, remember, is in smooth muscle cells at least what then leads to the commencing of the sliding filament mechanism of contraction. Okay, so you don't get the commencement of contraction. So that's how beta-2 adrenoreceptors would inhibit any contraction that was occurring within the smooth muscle cell, basically. Okay, so they would inhibit contraction of the smooth muscle cell, and hence they would cause the smooth muscle cell to adopt a relaxed state. Okay, right. However, beautiful as that and simple as though that pathway may be, basically to get this to actually happen, what you have to do is put on unphysiological levels of beta-2 adrenoreceptor agonists. Okay, so we're not really sure whether this pathway is actually physiological. We're sure that it occurs if you stimulate the beta-2 adrenergic receptors with more agonists than they would ever get, but we're not sure about its physiological significance. There is another pathway, basically, by which beta-2 adrenergic receptors have been it's been elucidated that they work, okay, and this will require a bit more discussion. Basically, it's a bit more of a complicated mechanism, but it does have good experimental evidence behind it, and I'll give some of that at the end. Okay, so basically, let's discuss this other pathway. So this may well be happening, okay, but we're not sure, okay. This next pathway, again, it's still a little bit contentious, but we certainly think it probably happens at physiological conditions. Okay, so here is our beta-2 adrenergic receptor here, okay, with the amino terminal extracellularly and the carboxylic acid terminus intracellularly. Now, when some ligand comes and binds to this beta-2 adrenergic receptor here, so this is our ligand, what will happen is the beta-2 adrenergic receptor will change conformation, and that change in conformation will be relayed to the intracellular loops, and it will then interact with the heterotrimeric GSG protein, and it will activate it to an alpha-S GTP subunit. Okay, so I'll just draw it straight out. So here we have our alpha-S subunit, which is now activated and has GTP bound, and then it will also have the beta-gamma subunit, which I'll draw over here maybe. So here's the beta and here's the gamma subunit. And of course the gamma subunit still has its prenyl group attaching it to the inner leaflet of the phospholipid bilayer, and also the alpha subunit has that myristic acid or palmitic acid molecule attaching it to the inner leaflet of the phospholipid bilayer. Now, basically, in this other model, the alpha S subunit with GTP bound goes and interacts directly with a different target to adenylyl cyclase. So we're now going to talk about this other target for the alpha S GTP subunit other than adenylyl cyclase. And basically, it's something called a big conductance potassium channel. Okay, and to discuss big conductance potassium channels, what we need to discuss is a type of potassium channel known as a family of potassium channels known as the calcium activated potassium channels. Okay, so we're now going to discuss the structure of these calcium activated potassium channels, and I'm sorry if I get this the wrong way around at any point. These are potassium channels which are activated by calcium. Okay, so they are calcium activated potassium channels. So this is the right name. If I mix up the potassium and the calcium, then please do correct it. Okay, right. So basically these are potassium channels which sit in the plasma membrane, and again they are tetramers. Okay, so they're not just one protein, they are instead 4 proteins all stuck together. Okay, so I'll draw this here. So there are 4 of these proteins stuck together like so. Okay, in a tetramer. Now, what would be beautifully simple would be if there was just one subunit for a calcium activated potassium channel called the calcium activated potassium channel subunit, and you just stuck this together 4 times, you got 4 of them, stuck them all together, and that gave you a calcium activated potassium channel. It's not that simple. Instead, there are 8 different subunits of calcium activated potassium channels. Okay, and these are grouped into 5 families. Okay, so we call them the KCA1 family, which is the calcium activated potassium channel 1 family, the KCA2 family, the KCA3 family, the KCA4 family, and then the KCA5 family. And as we'll see, KCA4 and KCA5, it's a bit of a misnomer. They shouldn't really be in the family of calcium activated potassium channels because they're not actually activated by calcium, but never mind, we'll come back to that in a moment. Okay, so let's look at the subunits which are grouped into each of these families. So in the KCA1 family, there is then the KCA 1.1 subunit, okay? In the KCA2 family, there are 3 subunits, okay? Whoops. There's the KCA 2.1 all the way down to KCA 2.3. So there's KCA 2.1, KCA 2.2, KCA 2.3. Okay, in the KCA3 family, there is again just one member of the KCA3 family, the KCA 3.1. In the KCA4 family, there are 2 members of this one, KCA 4.1 and KCA 4.2. And then finally, to take us up to 8, in the final family, there is just one, KCA 5.1. Okay, so 1, that takes us up to 4, 5, 6, 7, 8. Okay, so these are the 8 different subunits for calcium activated potassium channels. Now, basically, if you want to build a calcium activated potassium channel, these are the general rules. What you do is you pick a family, okay? So you'll pick one of these families, and then you can create either homotetramers and heterotetramers. As long as the heterotetramers, you pick subunits that are from the same family, basically. So you can't mix and match between different families. Okay, so you can make homotetramers and you can make heterotetramers. So homotetramers, again, means that you use the same subunit in all 4 slots. Heterotetramers mean that you mix and match, basically, so you'll have multiple different types of calcium activated potassium channel subunit in the same channel. Okay, but you don't generally mix between families. So if you're going to make a heterotetramer, you will make it out of subunits that are in the same family. Okay, right. So the other thing now to say is that if you make a calcium activated potassium channel out of subunits which are within this KCA1 family, and obviously there's only one of these, however it does have different splice variants, okay? So you'll always make a KCA 1.1 homotetramer here, but you could have different splice variants of this gene basically, okay? If you make a calcium activated potassium channel using this KCA 1.1 gene, then your calcium activated potassium channel is what's known as a big conductance potassium channel, okay? And for short, the big conductance potassium channels are abbreviated to BK, okay? So BK channels. So you'll see this called B for big conductance and then K for potassium, okay? And some people will then also put Ca2 plus at the bottom there, subscripted, to remind you that this is a calcium activated potassium channel, okay? And these channels are actually activated by two things. They're activated by voltage, okay, specifically by depolarization of the cell membrane, and they're also activated by calcium. So these are voltage and, whoops, missed the L, voltage and calcium sensitive. So if you've got a high concentration of calcium on the cytoplasmic side, that will open the potassium channel. If you've got a depolarization, that will also open the potassium channel, okay? So that's why they are in this family of calcium activated potassium channels, because they're activated to open by calcium. Okay, next one. If we instead build a calcium activated potassium channel from the second family's members, okay, then you end up with a channel that is called a small conductance potassium channel, and for short, small conductance potassium channels are abbreviated to SK. S for small conductance, K for potassium, and then some people will put Ca2 plus at the bottom. Now these small conductance potassium channels, these are not voltage sensitive. They are only calcium sensitive, so they're only activated to open by intracellular calcium, not by voltage. Okay, next family along, the KCA3 family. If you make a calcium activated potassium channel out of the genes which are in the KCA3 family, okay, and again it will have multiple splice variants, so you make a tetramer where all four subunits are made from this gene. They might not all be the same splice variant, however. It's called an intermediate conductance potassium channel, and for short this is abbreviated to IK. I for intermediate conductance, K for potassium, and again some people will put Ca2 plus at the bottom to remind you that it's calcium sensitive. Again, these IK channels are opened by intracellular calcium, okay, but not by voltage. So the big conductance potassium channel is the only one that's actually opened by voltage as well as calcium. Now, these final two families, they are put with these because of structural similarities, but they are not actually activated by calcium, so it's a bit of an unfortunate name and it might change in future. Okay, so if you make calcium activated potassium channel from these genes in the CA4 family, okay, you actually end up with a potassium channel that is activated by intracellular sodium and also intracellular chloride anions. Okay, so activated by too high sodium chloride concentration in the cytoplasm. Okay, so not by calcium at all. Similarly, if you make a calcium activated potassium channel out of the genes within the KCA5 family, you end up with a potassium channel which is opened by alkalization of the cytoplasm. Okay, so too high hydroxide anion concentrations. Okay, so alkalization activated. Right, so as I say, those final two families were put here because they have a similar structure basically, not because they were actually calcium activated. Okay, and it may well change in future. Right, so they are alkalization activated. Right, okay, so we are now going to look at the structure of the big conductance potassium channel in more detail, because this is the one we're going to be interested in, this one here. This is the one which the alpha S GTP subunit is going to interact with. Okay, so basically the big conductance potassium channel, if we look at the structure of a single subunit of the big conductance potassium channel now, so if we pull out one of these subunits of a big conductance potassium channel, and remember a big conductance potassium channel is one where you've used this gene 4 times basically. Yes, you might have different splice variants, but basically all of the different splice variants of this gene have the same membrane spanning topology. So let me now show you that membrane spanning topology. So basically, the amino terminus starts extracellularly, then you have the first membrane spanning alpha helix, the second, the third, the fourth, the fifth, the sixth, a P loop, and then the 7th here, and then the carboxylic acid terminus finishes intracellularly. Now, you will notice that it has a similar structure to things that we've seen before, except that it has this extra one here. So this first membrane spanning alpha helix is actually called the 0th membrane spanning alpha helix, and then the others are then labeled in the normal way. So this one is S1, S2, S3, S4, S5. This loop is then called the P loop, and then we have S6 over here. So this is actually called the 0th membrane spanning alpha helix. Okay, right, so you put 4 of these together and you then get one of these big conductance potassium channels. Now, what's going to happen is the alpha S GTP subunit is going to come and bind to this big conductance potassium channel, which you find in the membrane of the smooth muscle cells, and it triggers it to open basically. Okay, and what's going to happen if that big conductance potassium channel now opens? Well, let's draw this. Okay, so we'll have our membrane here, then we've got our alpha S GTP subunit here. So this is the alpha S subunit, and it's got GTP bound to it. Okay, like so. So it's in the activated state, and now it's going to come and bind to our big conductance potassium channel over here. Okay, so here it is. It's a tetramer of these KCa 1.1 subunits. Okay, there we go. And basically what's going to happen is this big conductance potassium channel is now going to open. Now we know what happens when you open a potassium channel. Usually potassium goes out. Let's make sure that we still understand this. So, the concentration of potassium intracellularly is usually around 155 millimolar. The concentration of potassium extracellularly is around 4 millimolar. We then need to know what the electrical potential difference across the cell membrane of a smooth muscle cell is. Generally it's around negative 60 millivolts. So the cytoplasm of a smooth muscle cell is generally 60 millivolts lower in electrical potential than the extracellular fluid. Now, basically that tells us that the concentration gradient favours the movement of potassium out, whilst the electrical gradient favours the movement of potassium in. However, this electrical gradient isn't strong enough to beat this concentration gradient. It will reduce the amount of potassium that flows out, but you'll still get a net movement of potassium out. Okay, so you are now moving positive charge out of the cell. Okay, so potassium ions are going from the intracellular compartment into the extracellular compartment. Because you're moving positive charge out of the intracellular compartment, the electrical potential of the intracellular compartment will go down. Because you're moving positive charge into the extracellular compartment, the electrical potential of the extracellular compartment will go up. That means that when we ask what's the electrical potential difference from extracellular to intracellular, it's going to become more negative because this one's getting higher and this one's getting lower. So the amount that you'll have to go down from here to here is going to get greater. So we are basically going to hyperpolarize the cell membrane. Now, why does the hyperpolarization of the smooth muscle cell membrane stop the smooth muscle cell from contracting? Because if you remember back to how the alpha-1 receptor stimulated contraction of smooth muscle cells, it didn't actually have much to do with the electrical potential difference across the cell membrane, because all we did was activate the phosphoinositide pathway, which produced IP3, which then triggered the calcium release from the ER membrane. Okay, but if you remember, and I'll get the paper back if you can't, if you remember, what we have generally in smooth muscle cells is these calcium waves, okay? And the IP3 didn't actually activate the IP3 receptors to open itself. Instead, it changed the conformation of the IP3 receptors so that they could now be stimulated to open by calcium. And if you remember, to set off the calcium wave, we had to have a source of initial calcium that set off the first IP3 receptor, which had IP3 bound, which then triggered off the cascade, basically. But where did that calcium come from originally? Well, it's still a bit of a mystery, but we think it comes from the extracellular fluid, and we think that it probably comes from a voltage-gated calcium channel, i.e. a calcium channel which will be opened by depolarization. Okay, if we're hyperpolarizing the membrane because of opening these big conductance potassium channels, then basically these voltage-gated calcium channels that let in this initial calcium will not open. Okay, and then you won't set off the initial, you won't get the calcium that sets off the initial calcium wave, basically. So you won't get the calcium waves in the cell if you hyperpolarize the cell membrane. At least this is the thought, basically. Okay, and if you don't get those calcium waves, then you don't get the calcium that's going to bind to the calmodulin, which will activate the myosin light chain kinase. Okay, and therefore you don't get the phosphorylation of the regulatory myosin light chain, and therefore you don't get the contraction of the contractile units within the smooth muscle cell. So those are the thoughts, the current thoughts for how the activation of beta-2 adrenoreceptors actually triggers the relaxation of smooth muscle cells. Okay, this one I think has more weight behind it now than the phosphorylation of the myosin light chain kinase. To get that to happen, I think you have to stimulate the beta-2 adrenoreceptors beyond what is physiologically relevant. Okay, whereas this occurs at physiologically relevant stimulation. Okay, and you do notice hyperpolarization of the cell membrane of smooth muscle cells when you stimulate the beta-2 adrenoreceptors. Moreover, this is the great piece of evidence behind this. Basically, you can block beta-2 adrenoreceptors from being able to cause relaxation of smooth muscle cells, and you can do this by blocking these big conductance potassium channels. Okay, so there are certain useful pharmacological agents which can be used to block big conductance potassium channels. Okay, so both of them come from scorpions. They're both toxins found in scorpions. Different types of scorpions. I don't know the names of the scorpions that actually produce each of these, but they're both found in scorpion venom. Okay, so the first one is called iberiotoxin. Okay, and iberiotoxin is often abbreviated to IBTX for short. Right, and basically iberiotoxin is a selective BK blocker, so it blocks the BK channels and blocks them from conducting potassium. Okay, so even if the alpha SGTP subunit comes and binds to the big conductance potassium channel, you will not get the big conductance potassium channel opening if you're subjecting your smooth muscle cell to iberiotoxin, and indeed that would then block the beta-2 adrenergic response, which is relaxation of the smooth muscle cell, if we're assuming that the relaxant response is produced by this hyperpolarization, which is produced by the big conductance potassium channel. Okay, and indeed you do find that iberiotoxin completely stops relaxation of smooth muscle cells induced by beta-2 adrenergic receptors. Okay, so this is a BK blocker, basically. Okay, there's another drug, okay, which is also found in scorpion venom, a different scorpion, and this is charybdotoxin. Okay, so charybdotoxin, And for short, charybdotoxin is often abbreviated to CHTX. Don't just abbreviate it to CTX, put the H there. If you just abbreviate it to CTX, people will think you're talking about cholerotoxin, whereas if you put the CH there, it's charybdotoxin. Okay, now this one isn't quite so selective for the big conductance potassium channel. It also blocks the intermediate conductance potassium channels as well. So it's an IK-BK blocker. But again, charybdotoxin will completely block the relaxing response of smooth muscle cells to beta-2 adrenergic agonists. Okay, so that's some quite powerful evidence in favour of this second pathway. Okay, so although this one's more complicated, it's potentially the right one, closer to the truth. Okay, right. So let's finally end with some drugs which are actually used pharmacologically, well, clinically rather. These ones are experimental drugs. They're useful experimentally. Of course, you do not use these clinically. Some clinical drugs which are used which relate to beta-2 receptors are salbutamol and salmeterol. Okay, so salbutamol and salmeterol are both beta-2, selective beta-2 agonists. Okay, which means that they bind to the beta-2 adrenergic receptor and they activate it. And they are both used in the treatment of asthma. Okay, salbutamol is generally used in emergencies. Okay, salmeterol is slightly longer lasting and is used more prophylactically. But basically, if you're having an asthmatic attack, then what happens is the bronchial smooth muscle cells that surround your airways start to contract and your airways narrow, and this means that you struggle to breathe. So what you do is you inhale salbutamol. Okay, so it's within one of those inhalers. You squirt aerosols of salbutamol into the airways, and this will go to the smooth muscle cells that surround the airways and cause relaxation and hopefully reopen the airway back up. So these are beta-2 agonists, and they're both used in the treatment of asthma. Okay, so that concludes our discussion of beta-2 adrenergic receptors. In the next video, we'll turn our attention to beta-3 adrenergic receptors.